welcome everyone. It is my great pleasure to welcome today Professor Christine de Rosiers from Université of Montreal in Canada. So Professor de Rosiers has an illustrious career. So she studied biochemistry at University of Ottawa in Canada with a bachelor until 1977. Then she did her master's and PhD in medical biochemistry at Laval University in Quebec until 1982. Went to Munich, the most beautiful city in Germany, for a postdoc until 1985. When she came, then she came back to Montreal as a postdoc, working in nutrition and metabolism until '89, and from '89 on, she is a professor. First at Université de Montréal in the Department of Nutrition as an assistant professor. Then she was promoted in '95 to associate level at the same department, and in 2001 became full professor at Université de Montréal. In 2004, she was promoted or changed her affiliation as full-time researcher and professor at the Montreal Heart Institute Research Center, where from 2009 she was leader in Metabolomics platform and of the Center of Excellence in Nutrition and member of the department's head advisory committee. She has multiple other professional memberships and honors, including founding member as well as now president of a nonprofit organization promoting collaboration and training in research on cardiac metabolism that is called the Society for Heart and Vascular Metabolism. So that means her research interests are really in the developments of methods to assess the dynamics of metabolism in intact organ. Specifically in the heart. She's also looking into specific signaling pathways in dystrophic and diabetic cardiomyopathy as potential targets for therapy, but also harnessing metabolomics for biomarker and therapy discovery in diseases. So her lab themes are therapeutic intervention on personal nutrition, including genetic diseases impacting mitochondria, including inborn errors of metabolism, but also mitochondrial fatty acid oxidation, lactic acidosis, inflammation of the GI tract, specifically also for regulatory actions of long-chain acylcarnitines that are linked to adverse cardiac events such as arrhythmia and heart failure. So while this is her bread and butter of her research, we asked her specifically to talk a little bit about peroxisomal lipid dyshomeostasis today, because that is one of the interests in our lab. So that's why we thought, we found a paper that she published and thought that would be very welcome to talk about that topic today. Thank you so much, Christine de Rosiers. The floor is yours.
Okay, so thank you so much for the invitation. It's such a pleasure to be, well, I would prefer to be there with you in person than virtually. As I said to others, it would be warmer, not because of the weather, because here we are below freezing, but because it would be nice to be with all of you. So just to start, I think I can share my screen. Is that it? Or
yes, that's correct. You should be able to share.
Yeah. So I think I'll do this, and then I want to remove the floating control. Okay, so do you see my pointer?
Yes, we do.
Yeah, okay, good. I see faces, but I don't see anybody, so I'm just making sure.
Yeah, that's harder.
Yeah, I know. Anyway, so yeah, I was asked to talk specifically about this the project behind this paper that was published in 2019 in GCI Insight. And I've put the first pages here just to underline to you that many people are involved in this paper, and all of them are important in the study. Mathieu Ruiz is the first author, and you'll be hearing about him. He was postdoc in my lab at the time, but he has now his own lab, and he's adjunct professor at University of Montreal. So I'll be also showing pictures so you can relate to these people. So without waiting, that will be a little bit my presentation objective and outline, because because you asked me to start specifically about this project, which is a special project. There's a history behind it. It's not as you say, my main bread and butter, but it's very close to my heart, and you will hear why in the subsequent slides. So what I will illustrate is the power and value for biomarker discovery of untargeted lipidomics in combination with other targeted approach. And these are things you know very well, and even much better than me, but also what can bring a robust study design in a small patient cohort, and also combining sample from human and mouse model of disease when you're studying a disease where there's not too many patients. So the outline will be, I'll just give you some background about my research interest, one slide, and tell you a little bit about our platform, which is much smaller than yours. And maybe we can say it's a little bit like a French restaurant, smaller, but people are very enthusiastic and want to contribute to the metabolomic field. And then come to the major part of the presentation, which will be this project about where we came to study peroxisomal lipid metabolism, and ending with the conclusion. So this is my slide. So today I'll be talking about my interest in inherited metabolic and mitochondrial disorder, but main bread and butter because I'm in the Montreal Art Institute. And just to let you know, we have double affiliation. So I am professor in the department, but my lab is in the Montreal Art Institute, which is about half an hour drive apart, because this is where in Research Institute, this is where we can have more access to bigger facility. So throughout my career, and as you could hear from Oliver, it has been many years, 25, 30 years. I've always been with the driving force for me has always been about studying metabolism. It's a passion. I'm a biochemist, but metabolism is my passion. And my let's say, hypothesis has always been that these metabolic alterations are early biomarker and actors in disease development and are target for therapy. And I've been working in other disease, like heart disease, heart failure. Heart failure is my main other project at this time, and one project also in inflammatory disease. And at the center of this has always been mass spectrometry and measuring metabolite level and flux, also using C13 label substrate and mass spectrometry. And with respect to models, initially I've been working a lot with mouse model, starting with rats even, and then mice and cell. But lately I've sort of switched my research more to human study, and been working then more on plasma sample or urine sample. So to talk about our platform, so as I said, it's a smaller group. We have our mission, it has been created in 2009 because there's been a major grant that it's an infrastructure grant that was given to our institute for research in biomarker. And there was like one section on imaging, one section on system biology, and the metabolomics. I was heading the metabolomic component. And that's where we were able to sort of grow in terms of what we were doing in measuring metabolites. So this is the group that is working at the platform, which is the stable full-time group. They all have background in either biochemistry and chemistry. So Bertrand Bouchard, Isabelle Robillard-Frank, Caroline Dano, And Annick Forêt is our project manager. And she's the one that has like the master in physics, master chemistry, specialty mass spectrometry. She has worked 10 years in a company in proteomics before joining our lab. So she has really the experience that we needed to develop further our capacity at the platform. And Mathieu Ruiz, who's the first author of the study I'll be talking about, is also now co-director with me, as I'm not getting any younger, so I need people with energy to sort of be able to drive. And so we have now access to 6 instruments, which I'm sure is much less than you, but anyway, we have 2 LC-QTOF, 2 LC triple quad, and 2 GCMS. And we have developed over the time at the beginning more targeted method on label and C13 label organic and amino acid, fatty acid by GCMS. Fatty acid profiling is a very popular method for many people. Acylcarnitine, bile acid, sugar phosphate, and the development of our method are mostly driven now by what people want. But let's say our biggest, I would say in French, I would say cheval de bataille, or say horse, the one that we've been sort of working more and like more in terms of what we're doing is the untargeted lipidomic workflow, for which now it's using the high resolution LC-QTOF, and let's say it can detect more than 1,500 features, depending on the type of biological tissue of major lipid classes. So overall, our platform, let's say through the years, I've been handling about 200 projects, and we work more on a collaboration basis. We had about 50 researchers from 20 institutions and across the world, in Quebec, but also Canada, United States, and Europe. And we've been publishing about 65 articles from these projects. So that's give you an idea. I'll just give you a little bit more information about our lipidomic, because I know you have a really nice workflow too, but it may bring some questions for the discussion. So we have always seen the lipidomics as a mean of enhancing biomarker discovery, because the lipids represent 75% of the plasma metabolome, and we know they display important biological function. When we first developed the method, that was a couple of years ago, at the beginning, most methods were not able to measure at the same time the glycerolipid and the glycerophospholipid, and they had problem having good recovery of glycerolipid, let's say full recovery. So our aim at that time was to be able to have all major classes and keeping at the same time a good linearity, good recovery, good reproducibility, knowing that they have we have a very large, wide variation in concentration. But more importantly, we've been working a lot on the method to be able to identify isomer and to be able to identify the acyl chain of glycerolipid, glycerophospholipid, and sphingolipid, because, well, just as an example, we knew that some of these, depending on the acyl side chain, we know that some of them are more anti-inflammatory as the 22,6, or others are more pro-inflammatory as the 24. So the way we designed our method was to have a maximum coverage of all those subclasses in a single sample, single instrument, because we were limited also in terms of instrument, and often because in our institute, it's a cardiology institute, a third center where we have a biobank, we were sort of asked to be able to do these analysis in a small volume of sample. So we can do all this in 100 microliter, one instrument. And well, we've validated our method on a human plasma, and Annick has been really the one who did most, well, with the others, but she's been the driving force. let's say we made the choice to have a long chromatogram because we have an 80-minute run, which is much longer than what you have. But we've made the decision at that time because we wanted to have good isomer separation. And this is an example of 4PC38.4, where we are able to separate them. And so this facilitates the data processing and the lipid identification. So overall, this shows the 2D map that we have with, let's say, an example with the human plasma, where we have 1,108, almost a little bit over 1,000 MS featured, and we have 16 lipid classes. Now we have increased the number of lipid classes that we can see. This is now 21, but where we did put a lot of effort is to make sure that we had good reproducibility. And also from the compound that we identified, we characterized many of them by MSMS, including acyl side chain, and found that close to 400 of them had relative standard deviation less than 30% in their linear range. So we've put a lot of time just to be sure, because since we knew we were going to do a lot of cohort sample, that this had to be, that was important. So just as an example of the proof of concept study that we conducted in 100 plasma samples from a biobank from LT subject, this volcano plot just showed to you the impact of sex. So it's expressed as fold change, male over female. This is for the lipid subclasses. And here we have a volcano which focus on the acyl chain. And so from the almost close to 800 feature, 205 were annotated using MSMS, and they are shown with different color. And what is immediately apparent is that men have more triglyceride and less sphingolipids, specifically sphingomyelin. And for the acyl chain, they have more acyl chain with 24 arachidonic acid and less, which is the red. And the blue here is 22,6 is docosahexaenoic acid, which is less. So these results were done as a proof of concept just to show, and they were in agreement with what had been published before. So in a way, they were showing that our method was able to do these measurements with precision. And other detail is in the paper. So this is for the background, just that you know what type of method we are using. And now to come to the main subject, which is more mitochondria in disease, just as a background in a nutshell. So at the basis, we were interested in mitochondria, the impact of mitochondrial dysfunction. And we know, let's say, if we do this in a nutshell, there's 4 points that maybe you want to remember. So mitochondrial dysfunction is increasingly recognized as a hallmark of rare and common age-associated chronic disease. If we talk about inherited mitochondrial disorder, it has a prevalence of 1 over 5,000, and it has a really wide spectrum of clinical manifestation, and in general, very high mortality rate in childhood. One aspect is that management and treatment of patients with this inherited mitochondrial disorder are really not specific, so they remain to be established. And there's a lot of research being done, and more recently, people are getting organized to try to tackle this problem together, because it is really a problem. Pathogenic mechanisms are also not, we know there's impaired oxidative phosphorylation for most of these diseases, but about those other biochemical consequences, possibly underlying clinical manifestation, much less is known, because most of the work has been done on the mitochondria, but less has been done to understand what else is not going well in these patients. So the Lee syndrome French-Canadian variant, which is the specific type of mitochondrial disorder, it's a monogenic mitochondrial disorder on which we worked on. So historically, number one. It has been, the first description has been in 1993, and it's prevalent in one region of Quebec, the province of Quebec in Canada, which is about 5-hour drive from Montreal, northeast, where the temperature is even colder, and you have to drive to a park where there's nothing, just moose. So, but this is a very, very nice region, but there is a concentration of people over there where the carrier rate for this disease is 1/23. In terms of biochemistry and genetics, it has been described as a tissue-specific defect in cytochrome oxidase, which is complex 4. And the mutation has been identified, the gene has been identified 10 years after the first clinical description as being LRPPRC, which is leucine-rich pentatricopeptide containing protein. And this gene in particular, there's one mutation which is present in all patients of the Quebec, and an additional mutation which is like a point mutation, and it's a compound heterozygous, but all patients have this A354V point mutation. Now, there's other mutation that has been described in people from China, also in England, and people coming from Turkey. So there are other places in the world now that this has been over in the past 5 years, but before only people, this disease was known only in Quebec. In terms of clinical manifestation, there's hepatic steatosis. Most patients die at a young age following severe acidotic crisis, and they have neurological involvement and multi-organ failure. So in terms of where the disease is most prevalent and where the tissue-specific defect in cytochrome oxidase is most prevalent is liver and brain, from what has been known from the autopsy. In terms of pathogenesis and treatment, it's like the other inherited mitochondrial disease. We know there's impaired oxidative phosphorylation. There's no treatment. And again, we don't know much about the other biochemical consequences, which could underlie clinical manifestation. So just one short, two little slide about the story behind this project, because why we did start this project, which is a bit apart from our main project, is because of this man, Pierre Lavoie. He's from this region where the disease is prevalent, and he lost 2 children from this disease. And after the loss of his 2 children, he decided to raise money for rare disease, and specifically this disease. So he started running in 1999, 650 kilometers in 24 hours, and started a regional event where people were doing biking to raise money. And let's say 16 years later, there's now like an event every year where they're across Quebec in 60 hours, and 1,000 persons are participating to this event. And there's also a smaller event for those who are less fit, which is 135 kilometers, which you have to do in 7 hours. And there is about 5,000 persons, which you can see here, which was the event in 2015. And it's a provincial event to which I participated, because this is me from this event in particular. So this has been really like one story about him wanting to raise money for, and he has now his foundation, which is Grand Défi Pierre Lavoie. And in terms of the project that we've been doing, in 2007, we created this consortium, the LSFC consortium. And this was the group in 2010 where we were in L'Abbé, which is this area where the disease is prevalent. This is Pierre with his little daughter. And this group was created to transpose the LR-PPRC gene mutation discovery into treatment for patients. And our goal, ultimate medical goal, was to predict the occurrence of crisis and find a reasonable treatment. This group has obtained, and I was the team leader when we obtained this grant from 2009 to 2014. And we were very active at the time, but the main granting institution, this emerging team grant, they decided to stop the program in 2015. So we were not able to renew this grant, but we've been sort of relying more on the Fondation Grand Défi to pursue at much smaller scale since 2015. And the group is also much smaller for those that are continuing the research. So that's about the story. But the global strategy that we had for our group, and fortunately we got this big grant to set up the project. So we did set up a biobank where we have cells, fibroblasts from the patient, we have blood, DNA, RNA, and fetal tissue. We were able to create some mouse model. Most of them were lethal, but we have one liver-specific knockout mice, which is viable, and which you will hear about in the talk. And also we had a group looking more on the functional consequences, where we had the molecular study and looking at mitochondrial function, doing metabolomics, metabolic fluxes on cells and animals, and a group looking at biomarker therapy and screening, where we would do the metabolomic. So what I'll be talking about is this part, a little bit of metabolomic in animal, and we'll be using this liver-specific knockout mice. So just prior to this study, where the lipidomic study, we had done a targeted metabolomic study, which was a case-controlled prospective metabolic profiling study. And this is the group of people that has been involved in this paper published in 2005. And the first author, Julie Thompson-Legault, is just here. But this is when we presented the result to the parent and patient in l'Abbé just before the publication. But what is important here is the study design, because this study design is the one that led to some sample collection that has been used for the GCI Insight paper. And I think this is an important, because we had only 9 patients. Those 9 patients, you were the only one alive. So that were the one to which we had access to. And so we knew that we had a very small N number. And so we decided to do a prospective matching of these patients with control, healthy patient genotype, which were matched for sex, age, BMI, and physical activity. Also, we were collecting fasting plasma and urine sample at home. So there was like and we were using a standardized protocol. So there was a 2-person, a nurse and a research assistant, going with their car, their dry ice, their centrifuge, to make sure that we were able to collect the sample correctly with this standardized protocol. And we were going at home because these patients are also fragile, and their parents are also, let's say, protective about what their child can go through. So anyway, from this, we did a profiling of 407 analytes, mostly hydrosoluble, using 2 platforms, one in Quebec and the other one was in Boston and led by Vamsi Muta. And we used several GCMS and LCMS methods and identified 45 markers reflecting major perturbation of mitochondrial metabolism, especially lipids, which were the acylcarnitine. And just to give you an idea, these were the metabolite, which are shown here, and each dot represent like a result from a patient. And it's expressed on patient versus control, for compound being increased or decreased. Same here with platform 2, and 9 were common to the 2 platforms, but just to let you show that the different color tells you whether it's metabolite linked to increase NADH, like beta-hydroxybutyrate or lactate, which were really expected. But for those lipids, beta oxidation are in blue, are all acyl carnitine, C18-1, C12, C16, are those that were reflecting, let's say, this dysregulated mitochondrial metabolism, especially lipids. So from there, this was the basis for the study for the GCI Insight paper. And there come Mathieu Ruiz, who's postdoc in my lab, and we have those samples still because we had collect them, aliquot them, and they were frozen at -80. And we were talking, and now our lipidomic method untargeted was developed. So we knew we could go further. And so we said, okay, we have rationale for probing further this lipid dysregulation. Basis being in LFC patient, we know that they have hepatic steatosis. They have high plasma level of acyl carnitine, which tells us that there are defects in mitochondrial fatty acid beta oxidation, which is expected because it's a mitochondrial disease that the complex 4 cytochrome oxidase is defective. But also in fibroblasts, we had made a study where we showed a greater susceptibility to palmitate-induced cytotoxicity. We had also characterized this liver-specific LRPPRC knockout mice. It had also hepatic steatosis and disrupted mitochondrial fatty acid oxidation. So with Mathieu, we asked, oh, what about the peroxisome? Because it's another organelle that plays a crucial role in lipid metabolism. It's highly enriched in liver, and little is known about the setting of mitochondrial dysfunction, the setting of peroxisomal metabolism in the mitochondrial dysfunction. Although we know that inherited peroxisomal disorder, like Zwillinger syndrome or Refsum disease, there's multiple mitochondrial alteration. And also, interestingly, the clinical manifestation in those inherited peroxisomal disorder, there's some similarity with the inherited mitochondrial disorder in that they have hepatic and neurological aberration and they die early in life. So with this incentive, we said, okay, let's see what we know about peroxisome. So if in 2 slides, I'll just summarize what like at the high level, what is the role of peroxisome in fatty acid metabolism first? Well, in fact, there are mostly there's a metabolic partnership between these 2 organelles. You know a lot about the mitochondria, which is here on the right side. And you know that the long chain fatty acid like C16, which is predominant in the diet, and also the oleic acid is also a long chain fatty acid predominant in the diet. They'll be beta oxidized in the mitochondria. This will generate reducing equivalent. You'll have oxidative phosphorylation, and this is important for the generation of ATP or energy. What about the peroxisome? Well, the peroxisome has a different way of handling fatty acid and also handle different type of fatty acid. So it will handle very long chain fatty acid, which can come from long chain fatty acid elongation or from the diet. It can also handle branched and odd chain fatty acid. And this can undergo either beta oxidation or alpha oxidation. And while it's undergoing beta oxidation, you will have shortened acyl-CoA chain, which through the carnitine shuttle can be further degraded in the mitochondria. And a big difference is that with peroxisome, you don't get ATP, but peroxide, hydrogen peroxide and heat as byproduct from this oxidation. Also, the peroxisome has a unique metabolic signature because there's not only these metabolism, which I've just talked to you about, which is more the catabolic side of the peroxisome for very long chain fatty acid or carboxylic fatty acid or branched chain odd fatty acid, but there's also an anabolic function. And for this, there's one step, which is the last step for the docosahexaenoic acid formation from the omega-3. The last step is the beta oxidation step, which is in the peroxisome. The second one is there's two steps for plasmalogen synthesis, which is obligatory and is going into the peroxisome before ending in the endoplasmic reticulum. And the last step is the conjugation of bile acid, which either to taurine or glycine, which is also taking place in the peroxisome. At least in inherited peroxisomal disorder, we know that there's a specific signature, which often is indicated here by the red, the red arrow. So you will see some indication that any of these processes can be perturbed. For example, for very long chain fatty acid, you'll see accumulating very long chain acyl carnitine like C26O. You'll see also some branch chain acyl carnitine or decreased plasmalogen or decreased DHA or decreased conjugated bile acid. So this is known from studies in the inherited peroxisomal disorder field, but not much when we started our study was known about inherited mitochondrial disease. So that's why our follow-up study with Lipidomic, we wanted to test the hypothesis that perturbation in lipid handling in mitochondrial disorder would go beyond what is known about impaired mitochondrial fatty acid beta oxidation and may involve also change in peroxisomal lipid metabolism, particularly in liver. And so Mathieu has been leading this project with two main collaborators who's been John Riou's lab, who he is a geneticist who's working in inflammatory bowel disease. And he has been generating this LRPPRC liver-specific LRPPRC knockout. And Jan Burel from University of Ottawa is our specialist in mitochondrial disease and assessing mitochondrial function. So we've used as study model this mice, but also sample from our patient from our first study and are untargeted and some targeted lipidomic using LCMS that we had established in the meantime. So these are the results that can be found in the paper too, but I'll be focusing on what is, let's say, the main result to bring the idea about the peroxisome. So this is the result. Here I'm just showing you the data processing because as you know, because you have also such workflow from the raw data, there's many steps in data processing to ensure that you obtain some very robust data. So at the end, from these over 2,000 features defined as mass over Z, retention time, and signal intensity, the filtered data consisted about a little bit over 1,000 entities. And so we applied correction to select the most discriminating feature. We had 21, which would discriminate the patient from the control. So this is plasma from our LSFC patient. And we identified 14 lipids by MSMS. Let's say our P corrected, our false discovery rate was less than 20%, and P was 0.0034. So these lipids, in fact, we didn't sort of add any expectation at the beginning, but this is the result we had. So we had the acyl carnitine, which we expected from our first study, 2 acyl carnitine, in fact, the 16-1, 18-1. We had a lot of glycerolipid, which with different chain, cholesterol ester, but the other class that we saw were ether lipids. All of those, well, they are shown here as plasmanil with the O, alkyl, because with our method, we are not yet able to, we're working on it, but we're not yet able to distinguish, let's say, to our satisfaction between plasmanil and plasminil, but these are the precursor of plasmalogen. So they were considered as the ether lipids, and we've been referring to them as plasmalogen. So to go further, once we had these 4 we said, okay, well, we need to be sure that these are, because this is an untargeted analysis. So we wanted to know, is this like a, maybe we saw this by chance or whatever. So we designed a targeted analysis with the LC typocrad, and we were able to see 21 ether lipid detected, of which 10 were significantly lower in our patient, as shown here by the gray bar are the patient, the white bar are the control, and with P varying from 0.05 and also after correction. From there, we said, okay, we have a change in plasmalogen. It's lower. So this may mean that the peroxisome is not functioning properly in terms of lipid metabolism. But is it sort of only plasmalogen synthesis? As indicate, I'm just showing here you a little reminder on the left, upper left. Is this only plasmalogen, or do we see changes in other pathways which are in the peroxisome? For example, can we see evidence for change in metabolism of very long chain fatty acid, branch chain fatty acid, DHA synthesis, or bile acid conjugation? So the first thing we did was a targeted profiling of acyl carnitine, because also we had a method for this using LC typocrad. And here, as shown as a Manhattan plot, you see all the acyl carnitine which are measured, which reflect either mitochondrial dysfunction in orange, or peroxisome, which is more like in purple and violet. And you can see at least that there's some that are tended to be higher but they didn't pass the P correction, but they had a P value of 0.05. And there was a major changes in those reflecting mitochondrial dysfunction, but nevertheless, there were some that were very close and higher in those reflecting peroxisomal dysfunction, which are the very long chain, the C26 or C24, or the odd chain, the C7 and a C9, although again, it didn't pass after correction. So we said, oh, well, we need to go further in this. So we went to look at not only at the bile acid. Here for the branch chain, I'm showing you just the results so you can see more the variation. So for the branch chain odd acyl carnitine with 7 carbon, you see that the increase was there. As for the very long chain, which we measure with also a quantitative method, there was no change which was significant. From the profiling of the bile acid, we could see that there was a decrease also in the conjugated bile acid with variation in unconjugated, but not significant, but the conjugated one, they were decreased. So this again was sort of pointing towards a change in the capacity of the peroxisome to perform lipid metabolism adequately. So from there, we said, okay, according to what we have access to in patient, which is the plasma, there seems to be like a change in the peroxisome and peroxisomal lipid metabolism, lower plasmalogen, change in acyl carnitine, which reflect the peroxisome and also the conjugation of bile acid. And we said, oh, can we learn more about this using the LRPPRC knockout mouse model, which is a hepatic specific. So this model had been characterized before in a paper in Human Molecular Genetics, and we had characterized that it has in the liver this loss of complex 4. It has also a loss of LRPPRC. It has also hepatomegaly, and also from the some lipid accumulation, but also infiltration of inflammatory cell and some vessel dilation from the H&E staining. So we use these mice and said, okay, let's look now at the plasma from these mice. So this again, and I'm showing here only the result for the peroxisomal lipid metabolism. We could discuss the other change if you want, but just to let you know that here we look at the ether lipids. 12 were identified. And again, if you look, these ether lipids, all of them were decreased in plasma of those mice. Again, is here and here, and both were, there were some which were phosphatidylcholine derived and phosphatidylethanolamine derived. So this, in terms of the plasma signature of these mice, it was consistent with what we had seen in patients, particularly for the ether lipid. In addition, we saw in mice a lower DHA level, which we didn't see in patients, but in a way, mice have higher plasma circulating DHA level than humans. So this is maybe the reason why it was a clearer effect in mice. But this was in addition, which point out even more to a peroxisomal lipid metabolism, which is disturbed. Then we went again to look at, okay, what about the liver? In the liver, we could see here, if you look at the acylcarnitine species, which are more on the right side of the slide, there's again this indices of mitochondrial dysfunction, because all these acylcarnitine, with the orange dot, short chain, medium chain, long chain, saturated, are increased. But here, those reflecting dysregulation of the peroxisome were really significantly increased, which include the C24, the C22. So those were significantly increased in the liver. And you can see here the box plot for these acylcarnitine 22, 20, and 26, and 26.1, which were increased. Also, we were able to see a decreased ratio in the bile acid, so conjugated over unconjugated bile acid, which were significant for most of them. So again, supporting the idea that there were perturbation of peroxisomal lipid metabolism. However, and this is a result that sometimes if you don't read the paper very, very, very carefully, you don't see this result, but I think it's important to put it forward. If you look in the liver further, we did look at the ether lipids, and we did do some molecular analysis of RNA and protein for plasmalogin synthesis. We look at the mRNA, and we looked also at markers of peroxisome biogenesis, transport process, and content, which include all those PEX gene and catalase, which were increased, and also increase in mRNA of enzyme involved in plasmalogen synthesis. Because in fact, in the liver, in contrast to plasma, plasmalogen level was all increased, as shown here again by those dark box plot for the knockout mice. And these are all the ones that we were able to measure. And this was a bit in contrast with what we had seen in plasma. And so in a way, that's why we were questioned to say, oh, what is the reason for this? And one thing we know about plasmalogen in liver is that in contrast to many other tissue like heart or brain, the level of plasmalogen in liver is very small. And it's regulated by FAR1, which has a negative regulation by plasmalogen. So the lower the level of plasmalogen, the higher the expression of FAR1. And this enzyme is located in the plasma membrane. So it has a site, regulatory site, which sends the level of plasmalogen. So we made the hypothesis that, okay, maybe in liver, it's the consequence that we see, that there is lower level close to the regulatory enzyme. And this is inducing expression of enzyme, the two enzymes that are involved in plasmalogen synthesis in the peroxisome. And they also, one of them at least, was increased significantly. So this was, in fact, a little bit supporting our hypothesis that maybe the liver reacts differently. Just to summarize this now, because just that you get an overview. So here, this slide reflects a bit the summary of the finding about the mitochondrial and peroxisomal pathway contributing to plasma lipidomic signature in LSFC patient and mice. So anything in blue would indicate a contribution from the mitochondria. So we saw the long chain acylcarnitine, medium chain, and hydroxyacylcarnitine being increased. We also saw increased triglyceride, which could be expected because of the dysfunctional beta oxidation due to the dysfunctional oxidative phosphorylation. As for the peroxisome, most likely it's contributing to the very long chain acylcarnitine increase, the odd chain acylcarnitine increase, lower DHA, lowered conjugated bile acid. And here I've indicated the expression of FAR1 and the enzyme involved in the plasmalogen precursor. And this the fact that the plasmalogen are decreased in plasma may be a signal for FAR1 to start to try to upregulate plasmalogen synthesis in the liver. So this is only a hypothesis. We don't have evidence for this, but it's at the moment our explanation. So just to end in terms of higher level summary of finding. So we use lipidomics in human and mouse carrying LRPPRC dependent mitochondrial dysfunction and identified the signature reflecting perturbation in lipid metabolism that goes beyond mitochondria and involve its close metabolic partner, the peroxisome. So what is the relevance? So there's many questions now. So what about the impact on other tissue? Is it only, do we see this only in plasma, or could it be seen in brain, heart, or other extra hepatic tissue? What is the mechanism? To have a follow-up on this, you could invite Mathieu Ruiz, because he has sort of been pursuing these questions as part of his own research project in his lab. As a group of the LSFC consortium, we are very much interested in the role of plasmalogen in LSFC pathophysiology, because we may link this to neurological involvement, because plasmalogen are known to be predominant in brain and in the myelin. Also, we're pursuing the fact that plasmalogen may be a target for therapeutic intervention. And beyond LSFC, if you look in the literature, well, we're not the only one talking about plasmalogen, because now it's becoming very popular. So what we see about plasmalogen have many biological roles in membrane integrity, particularly neuron as lipid mediator. They regulate cholesterol metabolism, immunomodulation, antioxidant. They're also involved in many other diseases beyond the one that we now have seen, the mitochondrial. We are adding the mitochondrial disorder, but it was known from the literature that the inherited peroxisomal disorder, which is expected, but also neurodegenerative like Alzheimer and Parkinson's disease. But the interest is there. There's some report about the potential of having a therapeutic intervention using alkylglycerol. And the scheme that had been proposed in terms of how this could be decreased has been mostly linked to obesity, where you would have mitochondrial dysfunction, oxidative stress, inflammation, which would bring this decrease in ether phospholipid and would be contributing to insulin resistance. It is our thinking that mitochondrial dysfunction can also be the start of all this story, because in our paper, this is not a question of obesity. Our mice are not obese. They only have a mitochondrial dysfunction in the liver, and they have hepatic steatosis and decreased plasmalogen. So this is maybe adding to our understanding of the mechanism. And I don't know if my time is out. I only have one last slide, two last slides. So do you have time for this one?
Yeah, sure.
Okay, so, well, this one may be of interest, because if you are interested in peroxisome, as I was saying, there's a lot of study going on, and I'm reading where they are reporting lower plasmalogen. I just read one about COVID-19, that low plasmalogen have been demonstrated in patients with COVID-19. But this is, pardon?
Where did you see that study?
I could send it to you. It's at Cell System 2021. They use a multi-omic. So I can send you the.
We would be very interested in that.
I just read it a few days ago. So, but it's a big paper. So you have to go through and digest it. I've read also your paper in cancer, which is very interesting too. Your paper in liver cancer. So anyway, in this case, it was a collaborative study in monogenic hereditary blood disorder, which is beta thalassemia. In patients. And in this case, this was very interesting. It's a collaboration with Gary Sweeney from University of York in Toronto. This is a volcano plot that we had. And well, interestingly, the signature of these patients was pretty similar to the one we saw with the mitochondrial disorder. A lot of triglyceride, as shown by those blue-green symbols. But also, and there were about 60 of these glycerolipids, mostly triglyceride, but also a lot of plasmalogen. The only thing they had in addition was the sphingolipids, sphingomyelin mostly. And here are the box plot of the most significant lipid, and the two plasmalogen are shown here, one in PE and one with ethanolamine and one in the PC. So that was interesting because it's a completely different disorder. It's a mutation in the globin gene. And so it's a blood disorder, but yet the signature was different. So maybe there are some common path. There's some hepatic problem in this disease too. So there may be a common path to the mechanism between these two diseases. So at the end, so this is my last slide before the acknowledgement. So just to remind you, well, we learned a lot of lessons from all this study in LSFC patients. The fact that we had a robust study design with our patient and prospective metabolic profiling, even though it was a small cohort, gave us nevertheless some robust results. The fact combining untargeted and targeted metabolomic, especially lipidomic, was really useful, but also combining human mouse model and also molecular analysis. And the fact that this enabled us to find this unexpected mechanism underlying lipid homeostasis, which you have a mitochondrial dysfunction, but now you have also an underlying additional peroxisomal dysfunction, particularly in lipid, and it implies the plasmalogen, but other pathway. And this may contribute to the LSFC pathophysiology. So for now, we're moving towards more identification of mechanism and testing the therapeutic intervention. So these are acknowledgements. The group in the team building day, see how cold it can be in Montreal sometime, but that was pre-COVID. We are now more Zoom apart. This was the group of consortium, which is not as big as before, but still some people are continuing in this study. And now the project is mostly financed by the Grand Défi Pierre Lavoie. So thank you very much. And those are all the founding agencies that helped building the platform and supported most of our work. Thank you.
Thank you so much. That was a great lecture. Thank you so much. We have a couple of questions in the chat room. Arpana, do you go through those?
Let me see. Yes, I can.
Either I could do it or you could do it. There's
You go ahead, Arpana.
Okay, great.
Okay.
Thank you for such a great talk. There's a couple questions in the chat room, and if there's more, please send them in and I'll monitor them. So one of the first questions is asking about your method specifically for if it can be used to quantify acylcarnitines, and if the acylcarnitines that you are looking at, if they're well separated in LCMS.
Okay, so for quantification, you can have, well, if it depends how many label standard you use. So if, well, we don't have label standard for all our acylcarnitine. We have, I don't remember exactly how many we have, and we are adding them as they become available. So some of them can be quantitative, but then you have to do also like a standard curve regularly, and often for screening, we do more semi-quantitative for profiling. And if then we do quantitative. For example, we did it for the C26 because we wanted to be sure. And for this one, we have a standard. So it can, and they say, are they well separated? Not all of them, because if you look, we have some that sometime in the paper, we give like a different symbol with just to hashtag more or less. And we say the number of carbon, but we're not sure about the structure. So then you could go through to make sure if you want, because it depends. You don't always want to have all of them. And often we offer like a smaller group, which reflect more mitochondrial dysfunction. But if the people want all of them, then if you really want them, you can work the method through to be sure that you have the right acylcarnitine.
Okay, yeah, that's definitely depends on how many standards you can get in terms of that question there.
Yeah.
There's
and
another.
Also, I think you have to verify with your standard curve. I think if this is what you want, you need to work 1 or 2 steps further with the method.
Yeah. There's another question from the same person. She says that lipids seem to play a role in many diseases compared to published literature. Do you find lipids that are differentially regulated in neurodevelopment disease versus neurodegenerative diseases?
Ooh. I can't answer that one because that's a good one, though. I don't, well, to my knowledge, I'm not sure well, lipidomics is also pretty, well, more recent than metabolomics in terms of technique. And I think it has exploded. There's been an explosion about lipidomic data in the past 3-4 years, and we'll learn more, but maybe, yeah, that could be a nice project.
Yeah.
Because you need to have the right model and to follow also through the development. So I don't have none of them come to my mind. Sorry.
It's totally okay. There was another question about gene knockout mice. We have a project with 30 gene knockout mice lines, and it was found that acylcarnitines and mitochondrial dysfunction related to the metabolites are commonly found in those 30 knockout mice genes. How would you, maybe from your experience, control these to make sure that those metabolites are specific to a specific knockout mutation?
Okay, just to make sure, when you say 30 gene knockout mouse line, you mean that each mouse line has 1 mutation, but you have 30 of them? Is that it?
Jenny, could you please?
Yes, they are just 1 gene knockout, gene mutation, not 2 or 3 combined.
Okay. And these gene mutation, are they, well, I think one thing I can tell you because we've been looking at acylcarnitine in many projects and many hereditary model, human and mice. It's a common finding to have for us anyway, to have acylcarnitine being upregulated at least in plasma, in other tissue, in liver often. And if you are interested, there's a nice paper which was published in Cell Metabolism where they show the importance of liver as a supplier of acylcarnitine in plasma. So it could be due to hepatic dysfunction, and it could also be due to any time you have a dysregulation of fatty acid oxidation, then you'll get change in those long chain acylcarnitine, C18, C16, C14. These are the ones that moves. If you have a longer chain, then this gives you, let's say you have a C21, C22, C24, then it gives you indication about the peroxisome. So it may mean that your gene, the gene that was knocked out, has an impact on those processes. So it may be direct or indirect, just like our LRPPRC. It's not a gene that affects directly the peroxisome. Well, at least we don't know, because we don't know of a direct action, but it could be direct or indirect. But it's a common finding, at least for us, in many models. So you may have, because one gene can have many impacts on many metabolic pathways. But the type of acylcarnitine can tell you whether it's mitochondria, peroxisome, or where exactly. Is it a long chain or hydroxyacylcarnitine? This may tells you that this the dehydrogenase step, or let's say each one tells you a different story about which enzyme is affected or which pathway is affected. Does this answer your question?
Kind of. So do you think the gene mouse knockout models can be used for interpretation of gene function on metabolism? Like
yes.
our model, or we have to specifically do the gene knockout and do cell analysis instead of the mouse model?
Oh, sorry, can you repeat your question? Sorry, I missed the...
Do you think the mouse model with gene knockout can be used for the analysis or interpretation of gene function on metabolism?
Okay, for human, let's say to
mouse model.
Are finding to human gene, you mean?
Yeah, also logs. Yeah.
Yeah, well, because in a way, I think there's are useful and informative, but there are differences too, which need to be taken into consideration. I think once you are aware, let's say, for example, in this one thing we've been going through in doing many study lately in mice, we have a very strong sex effect, which is much less, and even in disease, even when the mice is, let's say normally in patient or in healthy subject, we do see a sex effect. In disease, this sex effect is less apparent because the disease is so strong that it is if you look at the main discriminant lipid, it will be mostly the impact of the disease. But in mice, there's always a very strong sex effect, which may perturb your interpretation. So in a way, this is one example, but there are other examples of well, even let's say for us, the LRPPRC knock-in mice, which would model the disease, the mice doesn't survive. It's embryonic lethal, but the people survive. So there's a reason why the mouse model is not reproducing the human disease. I know of example, for example, the mice, I don't know if you heard about thermoneutrality lately. there's a lot of work that has been done saying that mice are raised in our animal facility and the temperature is not appropriate. It's too cold for it's not the thermoneutral temperature. And so that it will predispose the mice to certain disease that are, these mice are not normally perturbed, are not normally seen in, if you put them in thermoneutral temperature. On the other hand, these mice, if you don't they don't exercise, they don't miss their bus, they don't they're not exposed to COVID, they're not stressed. So there's many things that can influence the interpretation, but I think it's still useful and important because often we don't have other means to look at. And I think you just need to keep in mind that it's not a direct equation between the two.
Yeah, I agree. Thank you.
Oh, okay.
Great. Thanks, Jenny, for clarifying that. There's a question here, particularly related to gut microbiota. So can you imagine any downstream effects of the decrease in conjugated bile acids with LSFC? And could it be significant, for example, changes in the gut microbiota?
Why not? Is there any down? But it could be the reverse, because I think it could be also that I think in a way, with all what we know now of what can influence the gut microbiota, I cannot see, I'm sure that they have a different gut microbiota. We have not looked at this. And as much as the gut microbiota can influence the lipid signature or metabolite signature in blood, I think the reverse can be true too. So, but I don't know exactly, let's see, to pinpoint like a direct mechanism, I don't know.
Great. There's a question here from one of our senior scientists. Are plasmalogen species mentioned generally ether lipids, including alkyl bond ones and alkenyl bond ones? Plasmalogens refer to alkenyl bone ones, being a subclass of ether phospholipids.
Yes, yes, I did. Well, I mentioned rapidly, and I think I've, let's say I sort of say that I was misusing, in a sense, the word plasmalogen sometimes, because in our case, we are not able to directly assess whether we have plasmanil or plasmenil. We don't know. And so we said they were all plasmanil because we want to be secure on this, but it's and one objective of ourselves to be able to identify them one day. And we are collaborating with someone to try to learn more about this aspect. And I know that this has been always a challenge in mass spec. And there's been one study by Austrian group in Innsbruck, which has published in Analytical Chemistry that in 2020, and just were the first one to show how to differentiate the two groups. So this is a challenge, and I don't want to say that what we saw is plasmalogen, but these are lipids that are involved in plasmalogen synthesis, and they are ether phospholipids.
Okay, thank you. Oliver, you have a question?
So yeah, I have a couple of questions, of course, but I didn't want to dominate anything here. So first of all, peroxisomes can also be stained and looked at. Did you or anyone else look at, phenotyping of abundance, shape, other types of function for peroxisomes, for example, in those livers of those mice?
Well, beyond what we did as, let's say, the molecular characterization of RNA and protein expression, which were characteristic of the peroxisome, Jan Burel did some microscopy on the liver of these mice, but at the time he was looking mostly at the mitochondria. And yeah, I didn't think about it, but it's a good idea that maybe he could go back and check, because he didn't really, I think he was not looking for this in fact. But maybe he would have some information if he goes back. I don't know.
Yeah.
But
good
idea.
Second question I would have is following up your initial comment, actually, when you validated your method, you said, well, we clearly can distinguish men and women. Yay. And men and women are different in specific long-chain polyunsaturated fatty acids. And I think you said it was arachidonic higher in men, and I think you said DHA, as far as I remember, DHA higher in women.
Yeah.
So if DHA is also now affected by peroxisome function, would you see differences between men and women in either in female and male, either in your human studies for DHA-related metabolites, as well as in the mouse studies?
Good question. Well, the thing is with the human study, in human, there is the DHA level. Let's say when you do a fatty acid profile of all DHA, which are bound or to triglyceride and phospholipid, like total fatty acid profile, it's about like 1%, if I remember well. As with mouse, it's higher, so it's a higher signal, and I don't remember the right number for now. And we had only 4 male, 4 female as patients, and they were of different age. So it's true that we could go back and look whether there was a difference, but probably because we select, let's see, when you do untargeted, you impose and select a criteria and say, okay, I'll look at the most discriminant one. And it's true that it didn't go back and try to see whether we could see a difference in the DHA. Maybe it's there, but it didn't pass our threshold, as we did for, let's say, for example, in the liver of the mice, when we were using a pretty stringent threshold, we would not see that the ether lipids were higher in the LSFT model than in normal, but they were nevertheless with a good P and Q value, but we had chosen a stringent one because there were so many lipids, so we wanted to have those ones that are more discriminant. So to answer your question, we would have to go back in our data and see whether we can, maybe it's there, you're right.
Yeah. So you also mentioned at some point increased levels of triacylglycerides. Were these increased levels of triacylglycerides specific for very long chain fatty acids?
You in patient or in?
I think it was in patients, yes.
Yeah, the rise in triglyceride is more proeminent in patient than in mice, and I don't remember by art if it was because here, well, there's some like the 24, 22.4, 22.6.
Oh yeah.
There's some of them,
okay,
but it's not. Yeah.
Okay.
Not all of them.
No, I understand, and that's fine.
But in mice, we do see some special one.
Yeah, mice.
Like some unusual one, like 22, for example, things like that, which are not seen normally as often.
Yeah. So when we look at these fatty acids, these polyunsaturated fatty acids, they are precursors for a very important class of compounds that are lipid mediators called oxylipids.
Yeah.
Why didn't you look at oxylipids?
Yeah, we good question. Well, we thought about it, and we did look at oxylipin in another study, previous one with a colleague. We don't have, our method does not, we would have to set it up. We do see some of them like maresin or, you know, but not a lot of them. We cannot do a good profile of oxylipin. And we thought about it. I think it's more a question that we've not been set up to do it. And we were like, we're not a big group. So not enough people maybe said, okay, yeah, we need to do this because yeah, it would be a good, I agree, especially that in these oxylipin, there's some that are good and some pro-inflammatory and some are anti-inflammatory. So it would be a nice thing to do.
They have also a lot of more functions. So yeah,
I know.
I've seen in your LinkedIn profile that you're connected to John Newman, and he can talk about this for many, many hours, of course.
Of course, yes. I've not seen him for a while though.
Yeah, I know.
But I remember him from the Metabolomic meeting, and
yes.
Yeah, I agree with you. I think it's more a question, you know, it's a side project, and it's almost if we're doing this project with money in our drawer. And so
money in the drawer, that's always hard to have.
And so in a way, I think it has only not, if someone say, oh, you should do it, and we have an incentive, we say, yeah, you're right. I agree, we should do this.
Yeah. So you also showed a number of differences in oxidized lipids. Like I think there was at least one oxidized TG, and then I think an oxidized PC, and then you had all these hydroxylated acylcarnitines. You know, are these part of, what kind of oxidation or processes are these? Are these potentially coming from peroxide abundant?
You mean those ones?
Yeah, that was, yes, the epoxy TG you said here, exactly.
Yeah.
And lower there was a hydroxylated arachidonic.
Yeah.
Yeah,
yeah, you have a good eye.
And then there was also hydroxylated ceramide and so on.
Yeah,
so
here,
okay.
Here.
Yeah.
Well, honestly, I would love to discuss these with you because these are and we reanalyzed the LSFC sample with a more sensitive LC-Q-TOF, and we're able also to see those ones, the epoxy TG, and honestly, this is a project in itself, and we would love to be able to discuss these issues with someone else because we're sort of overwhelmed with whatever we can do, and we don't have enough. And we see those not in every group. Not all patients have those ones.
No, I mean, that's why it's after statistics, they pop up, right? Out of like thousands of features and so on, all of a sudden these things pop up, kind of implying that some type of either peroxidations or so might also play a role in either these beta thalassemia patients or maybe in other patients as well, right?
Yeah, these one, the beta thalassemia and the LSFC are the two study where we did see, and this one, it's the one which is the highest. You know, it's here, and that's why the box plot is the most. It's the one that has the highest fold change, and this one, the PC, is the one with the lowest fold change.
Yeah, so it's kind of like almost implying that it's not a generic feature of having more or less peroxides, but more of a almost like regulatory function or something like that, right?
Yeah, exactly. If you know more, I would be interested. Do you see them?
Well, we have Chase Stevens in our lab who is trying to hunt them down. So he's now conducting, he soon starts ozonations, and we will hope that we will find ozonation as, you know, as an environmental exposure to produce such epoxides, but we don't have no clue, of course.
Well,
we will also
if
try.
If interested, we could share. Yeah, we don't have anybody that is pursuing on this.
No, we are happy to collaborate, of course. So in our lab, it's Jay Stevens, who is kind of like at least looking a little more into these oxidated types of complex lipids, but we also try to really hamper or influence or use so on peroxisomes. You know, do you have, that's another question. Do you have any idea at all how to intentionally harm peroxisomes?
Intentionally what?
Harm, like
Harm.
Yeah, I mean, we know how to impact mitochondria, right? Or it's, you know, that's known. You have so many agents that would go and then harm the mitochondria.
Oh,
but we
you mean
want to do this. We
chemically?
want to make like a little dose response curves just to see, you know, how to handle peroxisomes.
Okay, you mean not using like genetic approach?
No,
or
no, yeah. So Jenny already said, like, you know, with mutants, it's all mutants are monsters, as we all agree, right? So you have so many downstream effects that it's so hard, right? I love chemical approaches, either, you know, environmental chemistry like ozone, or specific enzymes where you know like this is an inhibitor of that enzyme, or you know, yeah. Or maybe organelles or functions like you would have for oxidative phosphorylation in the mitochondria. We know exactly how we can manipulate it, and you know, we can fine tune it and then see the repercussions of that. So,
yeah.
So I am just now, we are just now starting to say, whoa, peroxisomes have been neglected by the community for far too long.
I agree.
You know, yeah, that's why I wanted you to talk about it, because you know, it's not like everybody talks about mitochondria for 50 years. It's okay.
Yeah.
They are important. We all agree. But
yeah.
What about the peroxisomes that have been neglected, right? So that's why we wanted to talk to you about it today.
Yeah, I'm thinking about, yeah, like let's say in terms of agent, yeah, fibrate, which will, let's say, stimulate peroxisomal proliferation, but something that will, I don't know, to chemically, I have to think about it.
Okay.
Well, if I will discuss this with Mathieu and see whether, because at this time I'm not, let's say, for example, mitochondria, you can always use so many things to inhibit respiration and things, but peroxisome, well, if you, because you can stimulate them. Let's say you have, you know, with phthanic acid that it's metabolized in the peroxisome, it will sort of be a challenge. But what would be the best way of challenging the peroxisome or inhibiting, or because I think it's adapting, and fibrate is one way, but it will probably do other things. I know, like Mathieu, what he does like he uses cells because he's interested at the impact in the liver, and he use siRNA to decrease like one enzyme and look at the impact. But that's a lot of work. And you may be interested. You could ask. He's doing some nice thing with his with these cells, but chemically. So are you using cell model or animal?
No, we are using hamsters.
Okay.
Yeah. Okay. Well, we are starting to use hamsters. So it's just like something we are starting right now. And the student who will do it is actually not even online right now because he has lectures to attend because he's a student. So that I kind of like try to, you know, ask for him in a way. Maybe also, you know, just thinking about what are future studies we can do in animal models. And in principle, you could do it in cell studies. I mean, I'm not against cell studies, you know, as long as you have viable cells and good cells and cells that are meaningful. I mean, it's
Especially in this case, like we know, for example, that hepatocyte liver is very different from brain or heart cell in terms of the plasmalogen. So
I was also interested in your finding that, you know, in the liver you had even higher plasmalogen levels than in the plasma you had lower. So it's like, okay, so then maybe the liver is not the ones that actually overproduce them and excrete them to the plasma, but some other organ, sorry.
But then it would have to be a secondary effect because the knockout is in the liver. So it would have to be a consequence. Yeah.
Oh, yeah. Okay.
But still, and also the fact that we don't know if there's not an exportation problem, because these, let's say, for example, at least in patients, we know that they have lower HDL level. So they could be a lipoprotein secretion problem, or like, we don't know if there is, because we know there's a link between, let's say, that plasmalogen regulate cholesterol metabolism, and cholesterol ester are decreased. So.
Yep. All right. So, well, with that, I cannot show you around on campus. I cannot give you a tour, you know, through the facilities, or have multiple meetings with other people like we all usually would do if this was an in-person meeting. Unless somebody else has questions right now.
If not, I'd like to share the information for the next talk.
Okay, so then thank you so much, Christine. And next talk will be...
Good luck with the COVID.